So we have scratched and clawed our way to the last chapter in the book. So congratulations, give yourselves a pat on the back. You are nearing the end. So this last lecture, we're going to talk about genetics and genetic diseases. It is chapter 25. So we will review some of those characteristics of genetics in human disease. We'll talk some about chromosomes and genes and gene expression. We'll also talk about genetic diseases, of course, and then the prevention and treatment of those diseases. Isn't it crazy to think that a human being can be made from a gene count only twice as great as that of a fly or a worm? If you've heard anything, read anything, or studied anything about genetics, you've probably heard of this man, Gregor Mendel. Gregor Mendel is famous for identifying what we now refer to as genes. Genetics began with Mendel more than 140 years ago when he identified something different with his pea plants. All right, and he identified that there are these little units called genes, and those genes have alleles, and those alleles, depending on how they're combined, are going to manifest them in different ways. So remember back to a time when we talked about the cell and we discussed DNA and how when DNA is inactive, it is in this tight coil that kind of resembles an X, right? And that is our entire genetic code, right? But this genetic code contains all of the instructions for all of the proteins that are made in our bodies. So a gene then is this unit or segment of our genetic code that when it is active, this chromosome strand unfurls, right? And it is going to allow the mRNA to transcribe that strand. Remember, it's going to go out into the cytoplasm, and the ribosome is going to come, and it's going to be translated into that protein or whatever molecule, proteinaceous molecule, which typically they're enzymes, and these enzymes serve to regulate the biochemistry of our body. So we could say that genes determine the structure and function of our body by producing a set of specific regulatory enzymes. So what does a gene look like? Well, here comes one now. Here it is. So we can see this tightly packed chromatin in the inactive state. It looks like this little X, or kind of like a little butterfly, is the chromosome. Now, when the body determines that it needs to make more stuff, then that strand is going to start to unfurl, and it's going to peel apart, right? It's a double helix. It's going to peel apart, and it's going to reveal a code. All right, and that code can get copied, and then that copy is what's eventually going to become the protein. Okay, so gene is a DNA segment that carries the genetic code, and then genes dictate the production of those enzymes, right? And lastly, when they're active, they're unfurled and available for coding. And when they're inactive, like when the cell is getting ready to replicate itself, then it is coiled and very compact. So the genome then is that collection of material in each typical cell of the human body. Now, in the typical genome, there are 46 individual chromosomes and then one special chromosome called the mitochondrial chromosome. Now in 2003, they started something called the Human Genome Project, and the goal of this was to map the entire human genome. Now, they thought that, and I would think so too, right? We're really complex creatures, so we must have a lot of genes. Well, they determined that it's not as much as we thought, about 19,000 genes, which is the smallest, among the smallest genomes of any animal that is out there. And approximately 2% of those genes code for proteins. The other 80% actually are like on-off switches. So they're going to tell genes to turn on and genes to turn off. And the rest is just, well, fluff. Parts of genes that we no longer need or we no longer use, or that have been kind of discarded for whatever reason. Extremely interesting. So here we are looking at an ideogram, and an ideogram is a simple cartoon that they use to represent chromosomes. And so all of these really colorful little X-looking structures, each one is called an ideogram. And this collection of all of these 46 chromosomes, we refer to that as a karyotype. And these are arranged in pairs by size. So you can see that chromosome 1 looks the largest, and then as we get all the way down to pair 22, they're extremely small. So this is the karyotype, all of these 46 chromosomes that you see here. So some anatomy quickly of the chromosome. The shorter segment of the chromosome is referred to as the P arm, and then the longer segment of the chromosome is referred to as the Q arm. And then they're joined together in the middle, remember, by that centromere. Here's some terminology that we need to familiarize ourselves with. The first one is genomics. Genomics is the study and analysis of the genome. And then proteomics. Proteomics is the analysis of the proteins encoded by the genome. Remember, we said about 2% for humans. All right, and then ideogram, which I already defined before on the slide previous to this, is the cartoon of a chromosome showing the centromere as a constriction, and the short segment being that P arm and the long segment being the Q arm. So we've had previous discussions about how genes are represented by their sequence of nucleotide bases. And remember, for DNA, that's adenine, cytosine, guanine, and thymine. And then for RNA, we replace thymine with uracil. Now, if you look at this top chart up here, this is actually a codon chart. So remember, a codon is 3 bases. And that codon, there's 64 of them, of possible combinations, and those codons code for 20 amino acids. So on the chart, you'll see the black type is the codons, and the blue type is the amino acid that they will code for. So as you might remember, cells divide by 2 processes. One is mitotic division or mitosis, and the other is meiotic division or meiosis, making more me's, right? So all the other cells, save our sex cells, divide by mitosis. That means their goal is to make an exact replica of themselves, a carbon copy of themselves, right? And that's what we want. But with meiosis, there are more than 8 million combinations possible. And this has to do with something called genetic variation. And we're going to get into that in a minute. But gametes divide by a different process so that we can get this genetic variation. And that's why, unless you're an identical twin, that you're going to have a unique genetic makeup because of this genetic variation. So each of these gametes has 23 chromosomes. We need 24 chromosomes to be whole, right? So the sperm has 23 and the ovum has 23, and when they join to make that zygote, now we have the 46 chromosomes to make the complete human genome. So here we're looking at the karyotype for the human. Again, all of these chromosomes. There are 23 pairs. If you notice, they're numbered 1 through 22, and then there's a separate set. Now, this separate set are the sex chromosomes, and they don't look like each other. They're not a match necessarily, but the other 22 pairs, we refer to them as autosomes. And autosomes are always going to appear to be nearly identical to one another. So here we've circled back to genetic variation, as I promised, and this genetic variation is an amazing thing that happens when we make sex cells. So what happens is when these sex cells go through meiosis, the chromosomes are going to shake themselves all up and redistribute themselves in a new and amazing combination. So that way each sperm and each egg is going to be genetically different from its other friends, right? So again, we call this genetic variation, and it's referred to, this process, the principle of independent assortment. So that applies to genes and or groups of genes. So during the process of meiosis, these chromosomes are going to do something called crossing over, and one part of that X is going to break in one area and another one is, and they're going to switch places, and who knows where that break is going to be, where that switch is going to take place. And so because of that, we have this this unique set of genetic codes. Now, sometimes there's a phenomenon that happens that's called genetic linkage. And this is when a whole group of codes stays together and does cross over as a single unit. So if you're wondering why you look like you do, that's heredity, right? We have certain genes, and these genes carry certain traits, half from mom and half from dad. And a combination of those is going to make us who we are, what our eye color is, what our hair color is, how tall we are, the whole 9 yards, right? So we know that each autosomal pair, right? We have those matching pairs of chromosomes, and on each of those chromosomes, there is a gene that says code for eye color that you got from dad. Now, its matching one is from mom, and on that same area, it has a gene that says code for eye color, okay? These codes or traits are referred to as alleles, right? Alleles are forms of a gene that are found at the same place on a chromosome. However, one might say code for blue eyes and the other might say code for brown eyes. This is where dominant and recessive genes come into play. So dominant genes are typically represented by a capital letter. So one trait that you get from one parent could be dominant or recessive, and a trait for eye color that you get from the other parent could be dominant or recessive. The dominant trait is typically the trait that is going to be manifested in the body, okay? And the difference between genotype and phenotype is this. Genotype is like these letters you see here, like big B, little b, big B, little b, little b. That's the genotype. The phenotype is what is actually manifested in the person. If you have one allele from mom, that's brown eyes, and one allele from dad, that's blue eyes, that means brown eyes is the dominant. You are going to get brown eyes. So big B, little b is your genotype, and brown eyes is going to be your phenotype. Now, alleles can also be heterozygous or homozygous. Hetero meaning they're different from each other. So that would be the big B and the little b. Homozygous means they're the same. So it could be a big B from mom and a big B from dad, or a little b from mom and a little b from dad. So like I said, recessive gene alleles are represented by lowercase letters, all right? And typically they don't show up in us unless each of our parents donated a recessive gene, recessive trait. Okay, so for example, blue eyes is recessive. So we're gonna say it's a little b. And if mom donated blue eyes trait, little b, and dad donated blue eye trait, little b, then you, my friend, are going to have blue eyes. But if mom donated brown eyes, which is represented by the capital B, dominant trait, and dad still donated that blue-eyed recessive gene, that dominant gene is going to give you brown eyes, even though you are carrying that recessive trait. So next we're going to talk about codominance, but before I do that, I just want to point out this four-square thing that we've seen a couple times so far. This four-square thing with all these letters around and inside is called a Punnet square. And this is how Mendel first started trying to map out if he had a pea plant that had these alleles and another pea plant that had these alleles, and he fertilized them together, what type of pea plant is he going to get? So these Punnet squares is what we use to determine what genotypes and phenotypes are going to be like. And we're going to walk through one of those, but I just wanted to point that out first. Now back to codominance. We already said that a dominant gene is represented by a capital letter. Well, what if two dominant genes are donated, one by mom and one by dad? Then what happens? Well, then we get something called codominance. And you can see here that black is dominant for chickens, apparently, and white is dominant for chickens also. And when you scramble those two together, you get this checkered looking chicken. It looks awesome. Okay, as promised, I'm going to walk you through one of these Punnet squares. And what we're using here is albinism. And albinism is a recessive trait. So it is going to be represented by lowercase a. And albinism is a lack of melanin pigment in the skin and eyes. So we're going to look at mom's genotype and phenotype, dad's genotype and phenotype, and then what the offspring's genotype and phenotype are going to be. Okay, so here we go. Now, mom, her genotype is represented by the letters on the left side of the Punnet square. And so her genotype is capital A, little a, and that means her phenotype is going to be normal skin color. So she's going to be called a carrier because she has the recessive trait, but it is masked by the dominant trait, by this capital A here. Okay, dad is represented across the top, and he has a genotype of little a, and to get a recessive trait manifested in the human, we need two recessive alleles. Okay, and so that's what we have here, little a. Dad is going to have albinism. So what if they decide that they're going to have a baby? All right, what is the chance that their baby is going to end up with this albinism? So let's see what happens. We're just going to multiply across by bringing letter on the left across and letters on the top down. Okay, so for our first square, we're going to bring the big A across and the little a down, and that's going to be our first genotype. So we're going to do the same thing on the top right, and we end up with the same genotype. Now let's look at the bottom left square. What do you think the genotype is going to be? If you said little a, you would be absolutely right. And what do you think about the last one? Yep, it's going to be little a as well. Okay, so what is this telling us? Well, this is telling us that 50% of the offspring are going to have a genotype of big A, little a, and the other 50% are going to have the genotype little a, little a. So what are they going to look like? Half of them, the ones that have big A, little a, are going to have normal appearing skin, right? Because the dominant trait is masking the recessive, but they're going to be carriers. Then the other 50% are going to have albinism because they're going to have 2 of those recessive alleles. Now, remember we said heterozygous and homozygous is also some terms that they use in genetics and inheritance. And so if we look at the big A, little a, those are different from each other, so that's called heterozygous. So 50% will be heterozygous for normal skin, and then the other half will be homozygous for albinism because the little a and the little a are the same. And that, in a nutshell, is a Punnett square. All right, so far we've been talking about inheritance in regards to autosomal traits. Now we're going to shift gears a little bit here and talk about sex-linked traits. These would be traits that are non-sexual traits that are attached to the sex chromosome. So we have the potential of an X chromosome or a Y chromosome. Now typically these sex-linked traits are attached to the X because really the Y only confirms or denies that you're going to have a girl, right? The sperm either carries an X or it carries a Y, and the mom is always going to donate an X because females have 2 X's. So mom's always going to donate an X, and then dad's going to be the deciding factor. If it donates another X, then it's a girl. If it donates a Y, then it's a boy. So typically these, like I said, are going to appear on the X chromosome, and we're going to take a look at what that looks like. Okay, so let's take a look at what a Punnett square would look like for a sex-linked trait. And here we're going to focus on color blindness. And color blindness is attached to the X chromosome. Like I said, Y chromosomes just determine sex for the most part. Those non-sexual traits that are attached to the sex chromosome are typically attached to the X. Okay, so let's look at what we have. Let's look at mom. Mom is across the top, and we know that because females have 2 X chromosomes. Okay, so mom's genotype is regular normal X, and then an X with the colorblind recessive trait. This means that she is a carrier, but she's going to be normal. She's going to have normal eyes, no color blindness, and this is because that regular X chromosome is masking that recessive trait. Okay, let's look at dad. Dad is a normal dad, right? There's nothing on the X is normal, the Y is normal. Okay, so let's see what happens if they were going to have an offspring. What's the chance that they're going to have a colorblind child? Okay, so we're going to do like we did before and move our letters across and down. And so this first left box, we get XX. And we're going to move to our next one. We're going to bring that normal X across and the colorblind X down. And then we will do our bottom left, bring the Y across, bring the X down, and then lastly, the Y across and the X with the colorblind trait down. Okay, so what is this telling us? This is what it's telling us. In regards to offspring, if these two peeps decided to have a child, there's a 25% chance that they're going to have a normal female and a 25% chance they're going to have a female that is a carrier. Okay, so something else we can see here is that there's a 50% chance they're going to have a female and a 50% chance they're going to have a male, right? But 25% chance normal female, 25% chance female that is a carrier. Again, eyesight's going to be normal with this female, but they are a carrier. They could potentially pass that on to their offspring if dad were to donate another X with the colorblind recessive gene. Now, bottom left, we have a normal male. That's awesome. And then bottom right, look what happens here. Mom donates that X with the recessive trait. So there is a 25% chance they're going to have a normal male and a 25% chance that they're going to have a colorblind male. Now, the reason that the color blindness is manifested in the male is because this Y is not masking the recessive trait. It won't hide it, okay? And this is the reason why men commonly have color blindness more than women do. I hope this helps kind of demystify how the whole sex-linked traits works. Okay, so we're going to spend a bit of time talking about genetic mutations. The word mutation by definition means change. So a genetic mutation is going to be a change in that genetic material. Now, sometimes these changes happen spontaneously, and they do and can cause bad things to happen in our bodies and have bad effects, but sometimes a spontaneous mutation can be good. For example, some humans have been found to form a gene mutation that causes tetrachromat, which means they have 4 types of cones that see color in their eyes instead of 3. So they can see these hyper colors better than a normal eye can. Another example of a beneficial mutation is natural immunity. And there have been some individuals found to have natural immunity to HIV, Ebola, malaria, and Alzheimer's even. And then another interesting one is a mutation that controls hypoxia. And they did this study of natives that lived in the Andes and the Himalayas, and they found that they had this mutated gene that allowed them to live in areas that had a lot lower oxygen concentration. So mutations can be good. Not all of them are bad, but those spontaneous ones are rare. Scientists also believe that a long time ago, no birds on the earth flew. And they attribute flying birds to a spontaneous mutation where a bird was born with no teeth. And because the bird had no teeth, it was then lighter and it was able to get off the ground and fly and escape its enemies. And so it went on to procreate and have more birds that could fly. More often than not, mutations are caused by some sort of exposure to our environment. And examples of that could be chemicals or radiation or viruses, and these are referred to as mutagens. So typically, these mutagens are what are causing the genetic mutations in our bodies. And sometimes they can be life-threatening, but other times, if they're mild, then they may persist in a population for many generations to come. So now we're going to talk about mechanisms of genetic disease. And the first one that's on our list is single gene diseases. So single gene diseases result from an individual mutant gene or a group of genes, and then these can be passed on from generation to generation to generation. On the other hand, sometimes it's not just a gene that is the factor in causing some sort of disease. It could be a combination of genes and our environment. And when that's the case, we use epigenetics to explain that. It's the science that describes how environmental factors might result in some sort of genetic trait that really can't be explained by genes alone. So chromosomal diseases result from chromosome breakage or from non-disjunction, and that means failure of a chromosome pair to separate during gamete formation. So there's two different types, trisomy and monosomy. And remember how we talked about the process of meiosis. In the process of meiosis, we have all of these chromosomes that are lining up at the epicenter, right, at the middle. And then these, chromatin stretches out to grab those chromosomes, and one chromatid is pulled in one direction and one chromatid is pulled in the other direction, and here we end up with equal numbers of chromosomes in each gamete. However, sometimes that doesn't occur and a chromosome fails to pull apart at that centromere, and this is called non-disjunction. So then we end up with a gamete that has two chromosomes, two autosomes instead of one. So when it joins with the other gamete, sperm or egg, then we have three autosomes rather than a pair. Now, monosomy is the opposite. Monosomy is the presence of only one autosome instead of a pair. And again, this can result from a gamete that was produced by non-disjunction. So you have, both of those chromatids were taken, and now you have a situation where you have a gamete that is missing one of those autosomes. So now when the two gametes join together, you only have one that was donated from a good gamete, and one is missing from the other gamete, and that is monosomy. Both monosomy and trisomy are typically fatal, and they also may be referred to as chromosomal genetic diseases, and they're not typically passed from generation to generation because typically the offspring die before they can reproduce. Cystic fibrosis is caused by a recessive gene in chromosome 7 that codes for a special protein called CF transmembrane conductance regulator. And this gene is needed to regulate the transfer of chloride and sodium across the membrane. But if there is an absence of this gene, or it is not working properly, then it is either not going to produce that protein, or it's going to produce an abnormal protein. And so what's going to happen is more chloride is going to be able to leave the cell, and when more chloride leaves the cell, what follows? Sodium, and then what follows sodium? Water, right? So then we're left with that thick, sticky, mucus-like substance that makes it really difficult to breathe. And so the respiratory system and also the gastrointestinal system are really going to struggle, and that can lead to obstruction, which can ultimately lead to death. Another example of single gene disease is phenylketonuria, PKU, and we have talked about that a little bit. It is also caused by a recessive gene, but this recessive gene damages the enzyme that is used to break down phenylalanine. And so since it is unable to be converted then we're not able to break down those amino acids that are in proteins. And so that is phenylalanine is going to accumulate in the blood, and it's going to start to destroy various tissues like the brain. Tay-Sachs disease is another example of single gene diseases, and it is common in Eastern European individuals as well as French Canadians. And Tay-Sachs disease is also a recessive condition, and what happens is because of this mutation, the body is not able to make an enzyme that helps to break down fats in the body. So now fats are going to be accumulating in the blood, in the brain, and then that can lead to severe mental disabilities and ultimately death, typically by the age of 4. Okay, so now we're going to talk about some epigenetic conditions, and the book does a really good job of making this confusing, and so I just want to kind of help simplify where the book talks about methyl groups and acetyl groups, and that some cancers or some mutations have been shown to stem from chemical markers known as methyl groups and acetyl groups. And these methyl groups and acetyl groups just very simply are kind of like the gatekeepers of transcription. So remember, transcription is that whole process of copying a segment of DNA to RNA, bringing it out into the cytoplasm, transcribing it, blah, 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 to make a protein, right? So these methyl groups are predominantly responsible for silencing that transcription. So they suppress transcription and they make it more difficult for transcription to occur. So they're more like the gate is closed. They close the gate. And then the acetyl groups, they make DNA more accessible to transcription. So they are more like the ones responsible for opening the gates, right? So transcription can occur. So if understanding that, it makes more sense that if we're having a situation where there is an overproduction of methyl groups, we're going to have too many genes silenced, right? And maybe those are genes that help fight cancers, like the p53 gene. So when we have too many methyl groups, that gate's going to stay locked, right? And if we don't have enough, maybe it's going to allow too much of a gene to be transcribed. And then the opposite effect is going to be with acetyl groups, because acetyl groups open the gate. If there's too much, the gates might stay open too long and let too much out. And if there's not enough acetyl groups, then the gates there's not going to be open long enough, right? So basically, this is how it contributes to cancers, by either silencing genes that aren't supposed to be silenced or transcribing genes that aren't supposed to be transcribed, okay? And one of the specific conditions associated with that is called Fragile X syndrome, or FXS, and it's associated with overmethylation. So too many methyl groups, right, like we just talked about, and that occurs on the X chromosome, and that can lead to developmental disabilities, can also cause like elongated structures, faces, foreheads, prominent jaws. You can see some of the images here of FXS. So I hope this helps make epigenetic conditions seem a little less confusing. So trisomy 21, Klinefelter syndrome, and Turner syndrome are all examples of chromosomal diseases. Trisomy 21 is the most well-known chromosomal disorder, and it produces a group of symptoms called Down syndrome. And it typically occurs in about 1 out of every 600 births, but as the female ages, that risk for having a baby with trisomy increases to about 1 in 80 births by the age of 40. Some characteristics we might see is mental disabilities as well as physical malformations. Then there's Klinefelter syndrome. It's another genetic disorder that results from that non-disjunction of chromosomes. And so we end up with 2 X's and a Y. So this disorder is occurring in males with that XXY pattern. And it's characterized by long legs, gynecomastia, low intelligence, small genitalia, sterility, and chronic pulmonary disease. Turner syndrome is sometimes referred to as XO syndrome, and it occurs in females with a single-sex chromosome X. So like many of the conditions described earlier, it is a result of non-disjunction during gamete formation. So there was a gamete somewhere that got 2 autosomes, and this one only got 1 or none. So when the 2 gametes fuse, there will only be 1 sex chromosome. Okay. So Turner syndrome is characterized by failure of the ovaries and other sex organs to mature, which typically causes sterility. Opposite of Klinefelter, it's going to result in a shorter stature, deformities of the neck, cardiovascular defects, and also learning disorders. So genetic counseling refers to professional consultations with families regarding genetic diseases. And so by compiling a familial history of as many generations back as they possibly can, they can help to give them a clear picture of what the probability might be of passing on some sort of genetic disease. This is 2 tools are used for this, pedigree charts and then the Punnet square that we talked about already. But this is why incest has a high probability of severe genetic mutations because typically each of us carries 12 to 14 serious genetic diseases, but they're recessive. So the more close our genetic code is, like a brother and sister, let's say, their tendency to have those same genetic mutations in their code is higher. So there is a higher chance that if they were to have a child, that those genetic mutations would be manifested in the child. And then that leads to those severe birth defects of the child. So disorders that involve trisomy or monosomy and broken chromosomes can be detected after a karyotype is produced. So here you can see an example of a karyotype. We looked at one earlier, but through the process of examining cells or through the process of amniocentesis, we can collect fetal tissue and examine those cells and determine if there is some sort of genetic malformation there. And then another way that they can test baby to see if baby has any genetic mutations is by chorionic villus sampling. And that involves the collection of the cells from outside of the chorion. And that can be collected by a tube that they insert through the cervical opening. Typically, most treatments for genetic diseases focus on treating the symptoms rather than attempting to cure the genetic disease. So gene therapy is something that we're starting to experiment with, and gene therapy is a way of manipulating certain genes to hopefully cure a genetic disease or a genetic problem. So the difference between gene replacement therapy and gene augmentation is that gene replacement therapy is used to genetically alter existing cells. Now, it doesn't replace the defective genes, but it inserts normal genes so that normal proteins can replace the abnormal ones or the missing proteins in the body's metabolic pathways. And one way that they're doing this is by using viruses as carriers. Gene augmentation is a little bit different in that rather than change existing cells, it adds genetically altered cells to the body. So we're adding altered cells, and then we're hoping those cells are going to start replicating and producing those proteins that need to be produced. A couple situations where we're using gene therapy is on a condition called Leber congenital amaurosis, LCA, and that's a form of blindness, and it's due to a defective chromosome. And so scientists are using viruses with a therapeutic gene that they have added in hopes that it will manufacture missing proteins in the eye, and that is showing signs of early promise. And another example is cystic fibrosis therapies, including putting therapeutic genes in cold viruses or sending something called plasmids, which are bacterial DNA rings that have been altered, and sending them into the respiratory tract. Now, there's also something else that they're trying called RNA interference, and RNA interference is a method of silencing particular genes so that they cannot encode for those proteins. So in the lab, it is working, and so it has potential as well. So that brings us to the end of our discussion on Chapter 25 about genetics and genetic diseases. I hope that you found it helpful and that it was beneficial to your learning. Thank you.